This is part 1 of transplantation immunology. So in this video we will be learning some initial aspects of transplantation immunology, and this particular study note is from EPG Pathshala site, Government of India, MHRD. So I would like to express my sincere gratitude to the development team for developing this note. Thank you so much. Then introduction. In immunological terms, transplantation refers to transferring of cells, tissues, or organs from one site to another. The tissue being transferred is called a graft. The receiving individual is host or recipient, and the one from whom graft is taken as donor. Transplantation is the only possible treatment in case of end-stage organ failure. It is not only an important clinical procedure but also involves and provides an important tool for understanding immune mechanisms. Transplantation rejection mechanisms displays cardinal features of an adaptive immune response like specificity, memory, self-non-self recognition. Self-non-self recognition. Rejection and shortage of donor organs impose major barriers in success of clinical transplantation. Different types of grafts and the associated chance of rejection are discussed in the present module, followed by various reasons and mechanisms to resolve the rejection issue. The current status of clinical transplantation and the special concept of immunological privileged sites and fetal transplantation are discussed in later part. We will be learning that later. Historical perspective. Let's have a historical perspective. The first case of transplantation was reported by Alex Carroll in 1908. He interchanged both the kidneys in 9 cats, and some of them could maintain the urinary output for 25 days. Though none of these cats survived, but it established that a transplanted organ could carry out normal function in the recipient. Peter Medawar made major contribution in the field of transplantation immunology. He was working on the burn patients during World War II. His work provided the basis for understanding the graft rejection from immunological point of view. Two major observations noticed that skin transplanted from one side to another within the same individual was readily accepted, whereas the one transplanted from relatives was rejected. Before Medawar, Panget's observation in Kansas City in 1932 made the underlying genetic basis for rejection apparent that skin allografts between family members tended to survive for longer than those between unrelated individuals. Additionally, J.B. Brown's critical demonstration in St. Louis in 1937 showed that monozygotic genetically identical twins accepted skin grafts from each other readily. Medawar also observed that rejection response was much faster and intense if a second transplantation from same donor is attempted. This observation were published by him in 1945, which laid the foundation of transplantation immunology, understanding the immunological basis of graft rejection. It was clear that in order to successfully transplant the organs and tissues in genetically non-identical individuals, the immune genetic barriers must be overcome. Alexis Carrel and Peter Medawar. The first successful human kidney transplant was carried out in 1954 in Boston between identical twins. In present scenarios, heart, lung, kidney, liver, pancreas, bone marrow, and cornea are transplanted with high degree of success in non-identical twins as well. Graft types. The grafts can be classified according to degree of variation between donor and recipient. Accordingly, the degree of immune response to a graft varies with the type of graft. Different types of transplant are noted. Graft type: xenograft, heterograft, heterogenic graft, xenogenic graft. Description: tissue transferred between individuals of different species. Kidney transplant from monkey to man. Vigorously rejected. Then next, allograft, homograft or allogenic graft. Tissue transferred between genetically different individuals of same species. Graft from one human being to another or from one mouse to another from different inbreed strain. Highly rejected. Allograft. Then isograft, syngraft, syngenic graft. Tissue transferred between genetically identical individuals. Transplantation between genetically identical individuals or monozygotic twins. Usually accepted. Then autograft, autologous graft. Self-tissue transfer from one site to another within the same individual. Skin transfer from normal healthy skin to burnt site. Usually accepted. Here you could see the pictorial depiction of this. Let me zoom this. Yeah. Allograft from one part of the body to another within the same individual, that's usually accepted. Then between allograft, between two different members of the same species but they are not genetically related, so probably the transfer is going to be a failure. Allograft, usually rejected. Then isograft, between genetically identical individuals like monozygotic twins, accepted. Xenograft, between individuals belonging to two different species, just vigorously rejected. Okay. Transplantation issue, rejection problem. With the advent of transplantation, it was seen that grafts received from other individuals in normal outbred populations are rejected. Studies in 1940s and 1950s showed that these rejection reactions are adaptive immune mediated, as they show the phenomenon of specificity and memory and are lymphocyte mediated. Various clinical and experimental evidence prove that graft rejection has all the hallmarks of all adaptive immune system reaction, as evidence that graft rejection to be adaptive immune reaction. Evidence: prior exposure to donor MHC molecule leads to accelerated graft rejection. Memory and specificity is seen in graft rejection. The ability to reject a graft rapidly can be transferred to a naive individual by lymphocytes from a sensitized individual. Rejection of graft is lymphocyte mediated. Inactivation or depletion of T lymphocytes by antibodies or drugs leads to reduced graft rejection. Graft rejection can be mediated by T lymphocytes, modified from basic immunology. Then attributes of specificity and memory in graft rejection. Specificity and memory properties of an immune response are always shown in graft rejection. Immunological memory of graft rejection can be understood by the fact that when an inbreed mouse of strain A is grafted second time with skin from strain B, the graft rejection takes lesser time than first time. Specificity of graft rejection is demonstrated when, along with the second time grafting from strain B to strain A, skin from strain C is also grafted in strain A at the same time. Rejection to strain B graft follows according to second set reaction and hence is faster, whereas rejection to strain C is slower, just like first set reaction. Hence, specificity as well as memory is already shown in immune response to graft rejection. Tempo of graft acceptance and rejection. Days post grafting, autograft acceptance, allograft rejection first set, allograft rejection second set. Days post grafting, allograft acceptance, allograft rejection first set, allograft rejection second set. 3 to 7 days revascularization, cellular infiltration of lymphocytes, monocytes, neutrophils, etc. 7 to 10 days healing, cellular infiltration of lymphocytes, monocytes, neutrophils, etc. Thrombosis and necrosis. Then 12 to 14 days resolution, thrombosis and necrosis. Role of T cells in graft rejection. The allograft rejection mechanism is T cell mediated. The nude mice that lack thymus and hence have no functional T lymphocyte are not able to reject allograft or xenograft. The allograft rejection mechanism is T cell mediated. The nude mice that lack thymus and hence have no functional T lymphocytes are not able to reject allograft or xenograft. Evren Meissen in 1950. The allograft rejection mechanism is T cell mediated. The nude mice that lack thymus and hence have no functional T lymphocytes are not able to reject allograft or xenograft. The first time proved using adaptive immune experiments that lymphocytes are responsible for transfer of allograft immunity instead of serum antibodies. Later studies implicated that the role of T lymphocytes in graft rejections. The experimental demonstration that T cells can transfer the allograft rejection is as follows. When the T cell obtained from spleen from an allograft primed mouse are transferred to an unprimed mouse of same strain, the recipient generates a second set rejection response to initial allograft from original allogenic strain. Naive mouse strain A grafted with skin from. So, naive mouse strain A grafted with skin from strain B. First set rejection, mouse strain A grafted with skin from strain B for second time. Second set rejection, naive mouse strain A grafted with skin from strain B. First skin graft, second set rejection 6 days after 6 days. T cell transfer allograft rejection. Both CD4 plus and CD8 plus T cells are involved in graft rejection. It was proved in a study where rate of graft rejection was measured after depleting one or both types of T cells using specific monoclonal antibodies. The control mice where no T cell were depleted took 15 days in graft rejection. The CD8 plus T cells depleted mice also took almost the same time as in control. Removal of CD4 plus T cells prolonged the rejection time from 15 to 30 days. Depletion of both CD4 plus and CD8 plus T cells resulted in further prolonged survival of graft tissue to 60 days. These experiments clearly demonstrated the involvement of both types of T cells in graft rejection and that the collaboration of both cell types results in more pronounced graft rejection response. Stages of cell-mediated graft rejection. Cell-mediated immune response to alloantigens present on the cells of grafted tissue or organ in the case of graft rejection. It involves cell-mediated cytotoxicity and delayed type hypersensitivity reactions. The process of graft rejection is divided into sensitization stage. In this phase, proliferation of lymphocytes in recipient take place against alloantigens present on the graft. The recognition of minor and major histocompatibility alloantigens by CD4 plus and CD8 plus T cells takes place followed by their proliferation. Dendritic cells serve as major antigen presenting cells in graft to activate host T helper cells. Most host APCs also migrate into graft tissue and process the foreign alloantigens. In some tissues and organs, APCs of graft also migrate to the lymph node of recipient and stimulate T cell immune activation. In some experimental cases, these donor APCs have been reported to induce tolerance in the recipient for their surface antigens. Endothelial cells and Langerhans cells are the other APCs besides dendritic cells that help in alloantigen presentation. Wait a minute. In this phase, sensitization phase, proliferation of lymphocytes in recipient take place against alloantigens present on the graft. The recognition of minor and major histocompatibility alloantigens by CD4 plus and CD8 plus T cells take place followed by their proliferation. Dendritic cells serve as a major antigen presenting cell in graft to activate host T helper cells. Host APCs also migrate into graft tissue and process the foreign alloantigens. In some tissues and organs, APCs of graft also migrate to lymph node of recipient and stimulate T cell immune activation. In some experimental case, these donor APCs have been reported to induce tolerance in the recipient for their surface antigens. Endothelial cells and Langerhans cells are the other APCs besides dendritic cells that help in alloantigen presentation. They amplify the population of T helper cells that induce various effector mechanisms for graft rejection. Then effector stage. In this stage, effector mechanisms take place which lead to destruction of graft tissue or organ. Cytotoxic T lymphocyte mediated cytotoxicity, delayed hypersensitivity are the major effector mechanisms besides antibody dependent cell mediated cytotoxicity and complement mediated lysis, which are less common mechanisms. Cell mediated graft rejection primarily shows influx of T cells and macrophages into the grafted tissue or organ. The foreign class 1 alloantigens on graft or alloantigenic peptides presented by dendritic cells lead to CTL mediated cytotoxic reaction. The cytokines IL-2, IL-gamma and IFN-gamma and TNF-beta have been shown to be key players in graft rejection. IL-2 helps in T cell proliferation and effector CTL production. IFN-gamma helps in influx of macrophages and delayed type hypersensitive reactions. TNF-beta helps in cytotoxic reactions in graft cells. Among other cytokines involved, IFN-gamma, alpha, IFN-beta, TNF-alpha and beta increase MHC class 1 expression, whereas IFN-gamma also increase MHC class 2 expression. The expansion of these cytokines is increased during graft rejection that leads to enhanced expression of class 1 and 2 MHC molecules in graft. IL-10 and TGF-beta are involved in promoting tolerance by mediating T regulatory function.